Hello, everyone. So it's really my great pleasure to introduce Dr. Robert Gatenby, or Bob. I've known Bob for a number of years now. We share a lot of interest in evolutionary cancer biology. In fact, he's really been one of the founders of the field and really a real leader in it. So without giving any dates, Bob did his undergraduate in physics at Princeton University, and you'll see how that carries over into the way he thinks. He's very mathematical minded. He's very much a mathematician and a physicist. But then he went on and got his MD degree from UPenn and went into the field of radiology. And he is still a radiologist. In fact, he's the chair of the radiology at the Moffitt Cancer Center. So he's had stints at Arizona State University and at Fox Chase in Pennsylvania. But he's been, I think, about 9 years at the Moffitt Cancer Center, where he founded the Department of Mathematical Oncology, which was really the first in the country. So this is the idea that you can use mathematical and computational modeling to better approach diseases like cancer, including clinical trials. And I think he's probably got, right now, he's going to tell you about a new approach to therapy that takes into account evolutionary dynamics, the complexity of cancer, heterogeneity, and most importantly, the microenvironment. And this has led to one clinical trial that he'll give you a little bit of insight on, as well as other ones that are in preparation. But I really think this has the possibility of changing the course of cancer therapy. So without further ado, Bob. Now, do you have the mic? Are you going to use this?
I'm supposed to use this. Okay. So thank you. Thank you for inviting me. It's really been a pleasure, and it's not common that I go to a place and wish I could spend more time at it, but this was uniquely so, and I enjoy meeting the many people that I've had an opportunity to meet. And thank you for the opportunity to speak. The Moffitt Cancer Center is actually the third largest cancer center in the United States. It's not well known. It's only 30 years old, but has grown at a remarkable rate. And this is the IMO, the Integrated Mathematical Oncology Department. It's about 9 years old. We actually have 9 applied mathematicians as faculty members, and we just hired an evolutionary biologist to join us. Really a serious commitment to both evolutionary dynamics and the application of mathematical methods. Just a word on how this began. I spent most of my career in Philadelphia, and if you live in Philadelphia, you know that Benjamin Franklin pretty much started everything. He actually had an interesting experience, which I think is worth mentioning. He was a great scientist, among other things, and he was interested in watching a lunar eclipse one evening in Philadelphia, and just as he was about to watch it, a nor'easter blew in. It turned out to be a very damaging and destructive storm, as nor'easters are if you have ever lived there. The name is because the wind comes from the northeast. And Franklin, like all scientists of his day, thought that the wind carried the storm. The wind carried the storm. And because it was a nor'easter, he then assumed that his brother in Boston would also not have been able to see the eclipse, and was startled to learn, he actually talks about this, that the storm arrived in Boston after the eclipse was over. So Franklin, actually to his credit, recognized that maybe the wind wasn't carrying the storm. Maybe it was more complex than that. Maybe it had to do with movement along barometric pressures. He actually came very close to the modern understanding of meteorology. But importantly, I think Franklin was among the first to recognize that applying intuitive linear thinking to a complex nonlinear system, although can be intuitively appealing, can also be dead wrong. And I think that when we talk about cancer as a complex dynamic system, that one of the advantages, the reason that you want to use evolutionary first principles and mathematical models is to get around that intuitive linear thinking and to accommodate the nonlinear dynamics that often dominate the cancer biology and treatment. So what do we learn from meteorology? Now, weather forecasting, now 250 years from that time, has become pretty mundane. How is that the case? So weather is a classic complex nonlinear system. How do we learn to forecast it? One is that we have good data sets. Second, we have Navier-Stokes equations, the first principles. And third, we have sophisticated and powerful computational methods. More importantly, I think, is that these are integrated. It's not just any data is obtained in any way you want to. It's actually data that's obtained to parameterize models. And this kind of integration of theory and data, I think, is the kind of thing that we are trying to do at Moffitt within this integrative mathematical oncology program. So here are the topics. Cancer is a complex dynamic system, integrating Darwinian dynamics into the genetic paradigm of cancer, exploiting the properties of complex dynamic systems, and applying evolutionary principles to therapy. Now, complex adaptive, for those that aren't sort of familiar with this, complex is because it has many parts. Adaptive is, or dynamic is because those parts change over time. And these interactions are typically nonlinear. So again, for the non-math people, if x gives you y and 2x gives you 2y and 3x gives you 3y, that's linear. And the human intuition is very, very good at that. But if x gives you y and 2x gives you minus 4y and 3x gives you plus 8y, that is nonlinear dynamics, and that's something that human intuition really cannot accommodate. Now, almost every time I show the weather analogy, there's some smartass in the room that says, yeah, but there's nothing you can do about weather. And this has been sort of popularized with this idea of the butterfly effect, meaning that a butterfly flapping its wings in Japan can give rise to a tornado in Texas. And this has to do with a couple of properties of complex systems. One is that they're extremely sensitive to initial conditions. And the other is there's a tendency to magnify some small perturbations. And so in general, this was used by Lorentz, who actually is the father of chaos theory, to indicate why you cannot control these systems. But more modern and applied mathematicians have taken a different view of this. And they've said, this tendency of complex systems to magnify small perturbations, if it's understood completely, if you understand the dynamics, you can exploit this tendency to steer a system in the direction that you want with the application of relatively small forces. And I'll talk about this. This is going to be a theme a little bit later. So to deal with this, Moffitt has embedded mathematicians in the Cancer Center. Cancer is a complex system. We try to integrate, similar to the physics paradigm, mathematical theory and sophisticated in vitro, in vivo, and clinical experiments. And this is a proposed cancer research cover showing this kind of the mathematical modeling below and experimental spheroid above. This is called the Collaboratorium, and we actually convinced Moffitt to devote a fair amount of space to a meeting area that is entirely informal. This is a place that can't be reserved. We have wall-to-wall blackboards for the mathematicians. We have PowerPoint projectors for the experimentalists, and this is where these groups meet to develop a common language. Evolution provides a unifying framework, and we are particularly interested in the role of imaging to understand the ecology of cancer, and I'll talk a little bit about that later. So fighting first principles. If you were to ask most people what are the first principles of cancer biology, it would probably be something like cancer is a disease of the genes. It's a remarkable number of papers actually start with that sentence, and is I would say, the dominant paradigm. And so the idea of somatic evolution is characteristically embedded in this paradigm. And so this is the Vogelgram. It's among the more famous diagrams in medicine, and basically, although it was published a long time ago, remains the kind of way people think about this. And that is that it is driven by mutations, that the mutations give rise to a change in the properties of the cells, and so you get a sequence of genetic changes that lead through a variety of premalignant states until you develop cancer and ultimately metastases. This is a very deterministic and linear process in this. For many reasons, it is, I think, unappealing as an evolutionary process, and I'll talk about that next. So most cancer biologists are surprised to learn that genetics is not an explicit component of Darwinian dynamics. And I want to show this in the context of This specimen, this is a ductal. Okay. 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 So this is a ductal carcinoma in situ, and it's stained for glucose transporter 1. Now you'll notice that this is all tumor growing into the duct. The interesting thing about this is that in general vessels don't penetrate the duct. So the vessels stay outside of the basement membrane, and all of the nutrients, all of the growth factors move into the duct by reaction to fusion kinetics. So as you get further and further from the basement membrane, you get lower and lower oxygen concentrations. And therefore, what you see is increasing amounts of glucose transporter because you have to use anaerobic metabolism, which requires greater glucose flux. My point is that the molecular properties of these cells is not random. It's not due to just a random mutation. It's there because the environmental selection forces are promoting it. It represents an adaptive strategy to the local environment. And this logical sequence of cause-effect, environmental selection forces, adaptive strategy, is Darwinian dynamics. Now, the molecular properties represent the mechanism of this, just as it does here. But you can perfectly well do evolution without any genetics. And so I want to sort of keep that in mind as we go forward. So what Darwin requires, if you ever read Darwin, I don't recommend it. It's like one long run-on sentence. So he didn't really come up with sort of general principles, but these are his general principles, and that is that the fodder of evolution is heritable variation in phenotypes. Okay, so evolution does not select genes; evolution selects phenotypes. Some of that heritable variation can be the expression of normal genes. Remember that the tumor has the entire human genome to select for strategies, including wound healing and developmental biology. So there's a lot of things that it can use that has nothing to do with mutations. Cancer cells uniquely, not like normal cells, but most normal cells, but uniquely competes for temporal and spatially heterogeneous environment. And fitness determines the proliferation. Those are the three Darwinian principles. One of the things that caught cancer biologists by surprise, but no evolutionary biologist, I think, would be surprised, is that there is a substantial spatial variation in the intratumoral molecular properties of cancer cells. On your left is the classic Erlinger paper. On the right is by Sotariva, by the Tavare group in PNAS. This is, I think, this is a renal cancer. This is a GBM. But the point is that there are lots of different subtypes. The molecular properties can vary considerably. Now, this is generally thought to be evidence of intratumoral evolution. But again, the classic description of this, the classic mechanism, is thought to just be accumulating mutations, hence this kind of diagram, or these kinds of diagrams, so that it's entirely mutation-driven. And I would argue that that's really not a Darwinian approach. And because remember that the spatial environments, the spatial selection forces of tumors is different. I showed you that in this ductal carcinoma in situ. Here's a GBM. Just look at the heterogeneity of blood flow. And so evolution by natural selection is governed by environmental selection forces interacting with the phenotypic properties. And so one reasonable hypothesis is that the spatial heterogeneity of the molecular properties of tumor cells isn't related directly to mutations, but in fact related to variations in the environmental selection forces driven, among other things, by variations in the blood flow. So to sort of beat this dead horse a little bit further, so what do genetics tell us and not tell us? So the fish above there is from the Gulf of Mexico. Swims freely, it's well known. So I need a student. I think I see some. So I always love the transition in facial expression from boredom to terror. Okay, so what's the fish below? Yes, I am looking at you, sorry. Okay, so yeah, so the top fish is just a regular fish. What's different about the fish below? Okay. Well, how about its pigment? No pigment? Okay. So what is it? I'm sorry? Okay. So what's the evolutionary dynamics? Okay. This is called a cave fish. Okay? And of course it lives in eternal darkness. And so it has no eyes, has no pigment. It's adapted to a cave. Now, for a long time, even Darwin was aware of cave fish, and for a long time people thought that they are the same species because all over the world they look exactly the same. It turns out there's 85 different species. And even if even the same species in different caves gets there by different molecular pathways. So I would question, so suppose you took all the cave fish in the world, ground them up, and did microarray. What would you learn from that? I would say you have a very complex data set, right? Because you've got 85 different species, even the same species. On the other hand, if you just look at the fish, it's pretty clear what happened, right? So this is classic convergent evolution. And my point here is that the cause effect is quite readily apparent. The molecular properties are behind that, underneath it, but often they are actually more difficult to understand than by simply looking at the fish. I also want to point out, did a blind fish find a cave? Right? Okay, so what's the order of how this happens? So, these fish begin to go into a cave. They begin to, not need the eyes and so forth. So it's again, remember, it's adapting to an environment, not the other way around. Now, one more question. Darwin was well aware of cave fish, and this was a question he asked. He said, "It's difficult to imagine that eyes, though useless, could in any way be injurious to animals living in the darkness. Why do they lose their eyes? They're not useful, but why lose them?" Right? Okay, so it's not contributing to their fitness, and in fact, there's a cost to it, right? Even the eyes, even in an intensely Darwinian environment where they're competing for substrate that's limited, the eyes cost something for them to make and maintain. The pigment costs something. There's a cost to that. And now, as we go on and think about cancer therapy, I want you to remember these costs. Okay, one more, and then I'm done. Okay, here's dandelions. This is in a field. These are about 30 centimeters high. They give rise to about 3 flowers a year. Here's a dandelion in your front lawn. Okay, this is only about 2 centimeters high, and it gives rise to a seed packet once a week. Okay, so we could do genetics on this, right? We could see how these two are different. Okay? Or we could think about what's the Darwinian dynamics. And so tell me, what's the Darwinian dynamic of this? It's what? Okay, but why? Why is it low and not high? These are the lawnmowers. They are adapted to the lawn mower. They stay low because otherwise the mower blades would injure them. Because most people do their lawn once a week, their seed packs are in sync with that. Again, my point is that the molecular data can give you information on how, but it's the Darwinian stuff that tells you why. Okay, so try to keep that. Okay, so here's some hypotheses. Intratumoral variation, intratumoral heterogeneity is due to variations in environmental selection forces and reflected local adaptive strategies. Okay. Tumors can co-opt highly plastic normal cells to optimize their environment. This is called niche engineering. So here's a question for you, and I don't have the answer to this, but I think it's worth thinking about. How many niches do you think are present in a tumor? How many species are present? It's surprising how, to an evolutionary biologist, this would be a very natural question. They figure out how many niches and species there are in a single pine tree, for example. And yet we don't ask that question in cancer. But we'll do it a little bit here. So we're going to assume for the moment that cancer cells can evolve. We're going to assume that normal cells do not. and that cancers are subject to classic evolutionary trade-offs like fecundity versus survivorship. So what do I mean by that? So fecundity is what field mice do. They breed a lot, they don't live very long. And fecundity is like what we do. We live a long time, but we don't breed very much. You can invest in one or the other, but in general, you can't invest in both. That's the trade-off that is classic in evolutionary dynamics, and there's a lot of others. So we began looking at this just very simply. Suppose we just take a classic Locke-Volterra evolution equation, and we start to say, well, what are just in that very simple kind of environment, what are the niches that are available to cancer? And we began to see then sort of an evolutionary separation occurring, and it has to do with how quickly the tumor cells can adapt from one phenotype to another. If they are classic generalists, meaning that they can move from phenotype to phenotype to phenotype depending on the environment, then you could have only one population. But as the cost and the speed of that variation changes, then you have to start branching out. And so we propose that there are at least two species that should occur in most cancers. And these are just, again, straight out of Locke-Volterra equations. One are engineers. These are K-selected, meaning they increase their carrying capacity. And we would propose that these maximize their population by angiogenesis. The others we've called pioneers. They invest resources not in making angiogenesis, in making blood vessels, but in invading. They are the Genghis Khans of the world. Genghis Khan's soldiers had 5 ponies, weapons, and no tents. They lived off the land. And these pioneers are the invasive guys. They go in, they acquire blood vessels from the adjacent normal tissue. They don't actually make it themselves. So these are selected. These are ones that are selected for rapidity of proliferation. So now we begin to think, so how do these segregate themselves? And one of the things that's clear is that you can't be an invasive guy unless you're adjacent to normal tissue. So we're already starting to develop a spatialness based on these evolutionary pathways. And so one of the things that we see is that acid production appears to be a niche construction pathway of the pioneer. So this is a tumor. This is in a window chamber. This is a stain for pH. And what you can see is that at the edge of the tumor, extending into the normal tissue, is a highly acidic environment because these tumor cells are making a lot of acid and it's diffusing along acid pH gradients into the normal tissue. What does that do? Well, we know that it breaks down extracellular matrix, so you actually begin to see significant tissue damage. Normal tissue is not it does not respond well to acidification, and you can see this loss of extracellular matrix immediately around the tumor edge, similar to corresponding to the pH gradient. Perhaps more importantly is that it creates a immunological sanctuary. It turns out that T cells are greatly inhibited by the presence of acid. So they essentially, even though they're extending beyond the tumor and they're out in the normal tissue, they actually are creating little halos for this. And we can show that this greatly reduces the immune attack on these cells. As molecular markers, we think that carbonic anhydrase 9 and 12 represent good ones. And there's an interesting story behind this because those of you who have worked with breast cancer know that these are used as prognostic indicators. CA9 is thought to be a poor prognostic sign. CA12 is thought to be a good prognostic sign. One of the things that we've learned is, in fact, CA9 and CA12 are present in the same tumor almost always. But where they are in the tumor reflects these two subpopulations. CA9, actually, so these are exofacial catalytic sites deeply rooted into the pH. CA9 tends to make a very acidic pH, whereas CA12 tends to maintain a relatively normal pH. And as you might imagine then, we've now started to look at this in people. What we find is that if we look at the tumor cells at the edge of the tumor versus the tumor cells in the core, there's a substantial difference. And I'll just show this. Here is CA9. This is the center of the tumor. Here's the edge. What we can see is that at the tumor edge has a lot more proliferation. This is the Ki67, a lot less, this is caspase, a lot less cell death, a lot more CA9, and somewhat more angiogenesis. So this is somewhat less angiogenesis. So these are the two niches that we think about, the invasive niche, the highly glycolytic invasive guys, the more slow-growing angiogenic guys. Now, this has the question is, can we use this to like the butterfly effect, push it in the direction that we want? And so we've actually now looked at tramp tumor. Tramps are mice that are genetically engineered to develop prostate cancer. It's near 100% penetrance. They typically have a tumor within about 12 weeks that you can see on ultrasound. They typically have metastatic disease by 30 weeks, and they typically die before the age of 1 year. What we have been able to do is to take from the same tumor cell populations that have entirely different strategies. And in this case, one is clearly an angiogenic one, and one is the engineer, one is the pioneer. So C2s are pioneers. They're highly glycolytic, highly invasive. They have high levels of CA9. They are extremely motile. The C3 cells are oxidative, they're non-invasive, and they're highly angiogenic in vivo. So the question is this: can we mathematically model these and look at the relative role of each tumor in the overall tumor growth, of each subpopulation in the overall tumor growth? And I am hoping this is going to work. So these are the models that we've looked at. We're going to look at the evolution of cancer cells. This is untreated, and what you'll see is that this is the phenotype. The phenotype. This is acid resistant. This is This is acid resistant. This is glycolytic. What you'll see is that tumor cells evolve in this direction. Let me just see if I can run this. Up and over. Now suppose we give higher doses. So now suppose that instead of just letting it evolve, we begin to try to perturb it. And what we do is we begin to add buffer. You remember that the acid-producing cells created an environment which is acidic, and therefore it's a niche construction. There's a cost to that, though. The cost is that they have to use the Warburg phenomenon, meaning that they're using anaerobic glycolysis even in the presence of oxygen, and that's an 18-fold decrease in the efficiency of ATP production. They also start to produce an environment in which it's very acidic, and it costs them a lot of energy to pump the ions out. So what we find is that if you begin to add buffer, that you can begin to, you actually tend to maintain this sort of low evolutionary rate. And in doing so, we can begin to change the way the tumor grows. And so this is the example. What we found, what the models predicted, was a small perturbation, in this case a 0.2 pHe unit difference in the extracellular pH, that small changes are sufficient to create a population transition in which you select against the invasive ones and for the slower growing angiogenic ones. Okay, does that make sense? Does it work? So we've done this in tramp mice, and what we found, as predicted by the models, that without any therapy at all, the C2 invasive cancer cells dominate the population. Can we reduce that with buffering? And so it turns out, let me see what this is. If we add 200 mole of sodium to the drinking water at age 4 weeks, so before the transition from intraductal tumor to invasive tumor, we select for the C3 population in the duct. And because they're non-invasive, they can't get out of the duct. So we effectively remove prostate cancer from the animals. They actually go to senescence without developing prostate cancer. They have lots of intraductal tumor, but you'll notice it's all intraductal. They cannot get out. So by making this small selection, we have a population phase transition, which allows us to see a dramatic difference in the outcome. We see similar changes if we give it late. Now we have a primary tumor involved, but what we want to know is when you've got small tumors that are forming metastatic disease, can you inhibit that acid production that's necessary for invasion, and as a result of that, reduce the formation of metastases? And this is just shown here. We basically all but eliminate the presence of metastatic disease using that, and we select for the C3 cells. We've now actually seen this several times in a variety of other conditions. We've actually looked at genetically modified breast cancers and found that we can do that same suppression of the primary tumor. We've now also made experimental tumors where we take MCF7 cells, which are ER positive, slowly growing and relatively angiogenic cells similar to the C3 cells, and MB231s, non-invasive triple negative, highly invasive triple negative, highly acidic populations, and we look at them together. And again, it's a purely experimental tumor, and what we find is that ordinarily with tap water, the MB231 cells dominate and the tumor grows very rapidly. If we add bicarbonate to the water, MCF7 cells dominate, and it becomes a much slower growing angiogenic tumor. So we're just using small perturbations to manipulate this. One of the more interesting things is that we can actually turn an MCF7 cell, which is generally an engineering type of cell, into a highly invasive, highly aggressive cells by simply adding a gene, the PMA gene, the proton, sorry, I can't remember the details of that, but basically produce a lot of acid. And so by doing that, we can actually take an MCF7 cell, which ordinarily does not metastasize, and make it a highly aggressive metastatic tumor. So again, turning it from an engineer to a pioneer. So I hope that's clear. This is the first time I've ever tried to present this, and I have a feeling I'm not being clear, but this is the basic idea. And so here's the summary so far. There are different species associated with distinct habitats in the tumors. There's an aggressive pioneer lineage characterized by CA9, high acid production, highly invasive, and highly motile. Less aggressive engineers tend to have near normal metabolism. They tend to live in relatively neutral pH, and they're highly angiogenic. Engineers can be converted to pioneers by simply transfecting a hydrogen pump. So again, it's relatively plastic. And the properties of a complex dynamic system allow you to steer the tumor into a trajectory that you would like to have using the relatively minimal application of biological force. Okay, so now I'm going to change topics. And we're going to talk about evolution and cancer therapy. This is a classic outcome from, this happens to be targeted therapy, but it could be any therapy in a person with metastatic lung cancer. This is a pretreatment CT scan. You can see the tumor extensively infiltrating the lungs. This is about 4 months following treatment. You can see there's been a substantial improvement. This is about 8 months later, and you can see that the tumor has regressed, has come back. So no matter how good your therapy, evolution generally defeats it. When we talk about personalized medicine, typically the focus is on the left and middle situation. So you want to see a target in the cancer that you can then treat and get a good response. I would argue that we need to be thinking, in addition, of the middle to right. We need to understand the evolutionary dynamics that lead to resistance and see if we can intervene on those. One of the problems is that the war on cancer has energized patients and politicians to expect a cure. You have President Obama, even my own institution, will today be the day, implies that any minute now we're going to find a cure for cancer, right? And then you can get clever and you can get self-serving. But the point is that we talk a lot about cures, and the focus on cancer therapy is arguably about curing. And so this is the classic Norton-Simon hypothesis, which dominates much of the thinking in cancer treatment today. The proposal was that you had to hit the tumor hard and fast. You had to give the highest dose possible in the shortest period of time possible. And so instead of the sort of zigzagging and the evolution of resistance, you would actually kill off the tumor, and that would be that. And this is, if you remember, I mentioned this idea of linear thinking. And this is a classic example, I think, of linear thinking. I mean, it makes intuitive sense. And it would be fine if it worked, but now 50 years later, we do this and we have a 5% or so mortality rate from many of the therapies that we give, but we're not really curing most cancers. So what's the problem with this high-dose density therapy? Because evolutionarily, it turns out that it's unwise. So let's assume that we're starting here and that each of these tumors represents a mixture of cells that are sensitive to therapy and cells that are resistant. And then we give high-dose drug. You essentially get rid of all the tumor cells that are sensitive, which at one point looks good because the tumor gets smaller. But the problem is that what's left behind are the resistant guys. And more than that, they have no competitors. They are totally free to proliferate unopposed. And so what happens, you give more therapy, they just get bigger, and eventually it returns. So this is actually a well-known evolutionary process called competitive release. This is the diamondback moth. And it's an interesting story, and I'll bore you with it for just a second. It was actually first found in the United States in 1854 in Illinois. It eats cabbage, which in my mind makes it a pretty good bug, but to farmers it's actually not very popular. And so this has been followed now for over 160 years. And so this is a well-known, well-documented phenomenon. Since its first discovery, over that 160 years, it has been bombarded with every insecticide ever developed. And what's happened? It's now spread throughout North America. It's in Europe. It's in Asia. It's in Australia. It is spread throughout the world, really. And since 1988, the moth has been reported to be resistant to all known insecticides. So I would argue that this is actually rather similar to a cancer, that we need to learn from this, what's now the policy in the United States, which is called integrated pest management. But the point is that if you were a farmer and you had a diamondback moth, what you would be instructed to do, first of all, you could go to a computer model and it would tell you how to do this, but secondly, you would be instructed to give only small doses of the insecticide. Just knock the thing down a little bit. But if you try to eradicate it, all you'll do is make for a resistant population. And that's actually forbidden by law in the United States. And so here's the problem. And I'm going to show, now suppose this top here is a phenotypic distribution related to sensitivity to a drug. And this is certainly not unreasonable. Now, I'm going to assume that we give one pulse of drug, and you'll see this happen. Okay, here we go. And you'll see a pulsar drug come up here, there, okay? So what happened? It was great. Really got rid of almost everything. But because there's a phenotypic tail, there's always going to be, at the edge of this, some cells that are resistant. They can be resistant because they're phenotypically resistant. They can be resistant because they're in some sort of sheltered area where the blood flow didn't bring a lot of the therapy. They can be there because mesenchymal cells, and they have found a partnership. But whatever it is, it's really hard to eliminate a large heterogeneous population with a single drug or even a few drugs. Okay, now the therapy is going to be withdrawn. And watch what happens. First of all, it grows very, very fast because this population, which was a minor population at the beginning, it was a small one in the broad distribution, has now become the major distribution. It is proliferated very rapidly. Okay, now, watch as we continue to let it go in the absence of therapy. And you'll see what's happening is that the phenotypic distribution is moving from right to left. Okay? It's going back to where it was. Okay, and why is that? If you remember the quote from Darwin, why do cavefish lose their eyes? It's because there's a cost to it. It's a clear advantage when you have to see, but when you don't, then it's a disadvantage. And so what's happening here is that there is a cost of resistance. It may not be very large, but it's sufficient that if you're not selecting for it, you will tend to move back to the wild type. Okay. So the best analogy I've ever heard was when I was a guest in a German institute. The host said, it's like an umbrella. When it's raining or snowing, it's necessary. But when it's not, it's a burden. Now, the burden isn't big, right? It's not much. But if you ask these people to run a race, if they were in an intensely Darwinian environment where they're competing with each other every minute of the day, then the person that was carrying the umbrella would have a disadvantage, again, in that very intensely competitive environment. So can we exploit this? And so the idea here is that the cost of resistance is always going to be present. It's not necessarily well documented. In fact, it can be hard to identify. But whatever it is, it requires resources to be diverted from proliferation. And there's always a cost. And we can see this if we take, these are MCF7 cells that have become resistant to doxorubicin because of highly uprated P-glycoprotein. If you put them in a culture dish with doxorubicin, the resistant guys always win. But if you put them together with no chemotherapy, the sensitive guys drive the resistant ones to extinction. And so this is adaptive therapy. This is the idea of how can we exploit these evolutionary dynamics to improve therapy. What you have to do is get rid of the concept that you're going to cure the patient. You're going to try to cure the patient. That's your goal. Now, if that's a possible outcome, then obviously you shouldn't be doing adaptive therapy. But this is only to be used in patients who have a fatal disease. The idea then is don't treat as though you could cure them. Don't give them high doses. In fact, give them just enough treatment to knock the tumor down. Now, you're going to get rid of some of the sensitive cells, but you leave some behind, and you explicitly do that. You, of course, have the resistant guys. You then withdraw therapy. You stop treating altogether. And if you remember the way that curve went, what happens is in the absence of therapy, the tumor will begin to grow back again. But because there's no selection for resistance, the phenotypic, the fitness advantage of the sensitive cells allow them to proliferate at the expense of the resistant ones. So at the end of this cycle, the bad news is the tumor is back. But the good news is it's really close to what it was before. And you can treat again. You can just keep treating. So the goal here then is to maintain enough cells, sufficient number of cells that are sensitive, and let them control the cells that are resistant, the ones you can control. Okay. Now, this was actually developed initially from models, actually. It was actually entirely developed mathematically, but then sort of converted into this sort of evolutionary paradigm. And it's now been modeled extensively, and now we've actually used it successfully in a variety of animal experiments in both ovarian and breast cancer. So we started our first clinical trial almost 2 years ago. We used prostate cancer. A lot of famous people get prostate cancer. The good news is that although it's fairly common, most people are cured. And the bad news is if you get metastatic prostate cancer, it's a nasty disease, and basically everybody dies. The prostate cancer is intimately associated with androgen. Nearly all of the initial tumors are sensitive to androgen deprivation therapy, and that causes a decline in their serum marker, which is PSA, typically for a year or two, and then they progress. About 2/3 of the time, when they progress, it's because they're making their own androgen, and so that pathway is actually the target for abiraterone. So this is the same idea as so androgen is upregulated through this cytochrome P450. And it produces androgen both for the cells of this autocrine loop, but it also produces it as a common good. It seems to spill into the microenvironment of the tumor where it can be available for other cells to use, and they are called cheaters because they're using the common good without actually paying the price for it. And so this is how we approach this mathematically. We defined 3 species. T plus cells are the ones that require exogenous testosterone. TP cells are the ones that make testosterone. And the T minus cells are independent of testosterone. These are the bad guys. These are the guys we want to prevent from proliferating. Now, again, there's an interesting dynamic because the testosterone produced by the TP cells can serve as a public good and permits cheating by the dynamics by the T plus cells, meaning that the cells that were treated originally with androgen deprivation, if there are any left, can come back and become part of these dynamics. This is the model that was used and is what we used to design the protocol. So we've used what we have. We then use an inverse problem effect. Basically, what we say is that if we want to estimate the relative fitness of each of these populations, we can't independently know that through direct experiments, but we can infer it by their relative abundance in the tumor. In general, although there can be exceptions, that the greater the population, the greater their fitness, so that we can rank them according to the pre-treatment biopsies. What we've looked at here is abiraterone. About 62% of men with metastatic castrate-resistant prostate cancer respond to abiraterone. Progression-free survival ranges from 6 to 11 months. And this is our model. These are the subpopulations. What we propose is if you give high doses of abiraterone, the PSA will decline. You will eliminate the TP and T plus cells, but the T minus cells will come back, and eventually this will be the recurrence. So this is competitive release. This is our proposed target. We stop at 50% of the PSA. We withdraw therapy. We let it grow back. We try to recapitulate the initial tumor, treat it again, let it grow back. This is the protocol, which really says the same thing. Here's some interesting, we didn't have a lot of biopsy material, but we had some, and what we found was that the 3 populations that we thought were present were in fact seen on this, and we can't even estimate their subpopulations. So we think that this is something that we should be doing more of. It was only available in 3 of the patients, but we were confident that we were seeing the subpopulations that we thought existed. We now actually have we have about 15 patients. At the first 10, what we've seen is the cycles that were predicted. You can see this is PSA. They're treated. They go down. They're stopped. So we have this kind of cycles. What's interesting is that how variable these cycles could be. So this individual, it's a little hard to see these dates, received 3 treatments in a year. This person has received 2 treatments in about 2 years. So it can be highly variable. So far, this is 10 of the 11 patients remain in control. They have been on trial for an average of 21 months, which is significantly better than the PFS that's reported for the best PFS that was reported in the New England Journal. We haven't reached the PFS. And what's interesting is that because of this up and down kind of treatment, they've only received 25% of the dose of abiraterone that they would have received under standard of care. So future directions. One of the interesting things is that what the model suggests is counterintuitive. We're always trying to increase the dose. And even as I present this sometime, I'll often get the response, okay, great, you've got a lot of dose that you can go up for. But in fact, the model suggests that what we should do is go down on the dose. And then in fact, what's happening is that every time the PSA goes down to a nadir, if it's 50%, you get a little bit of increase in the T-minus cells. So each time you do that, each time it's getting bigger and bigger. So ultimately, it will fail. Ultimately, it will be too large to control. Now, at its best, the best outcome will be 20 cycles. Now, if each cycle is between 4 months and a year, it's still a lot of time. But what the model says is that if instead of going down 50%, you just go down 20%. So now you get a lesser increase in the T-minus cells. That if you do that, the models predict that we can indefinitely control these tumors. There is a second, recently there have been some articles where they've added a cytotoxic drug, docetaxel. This is an example of that. Now, docetaxel is a cytospecific drug, and it's typically added with abiraterone, so they're given together. So again, the idea is that you just kill more cancer cells. And we don't think that's right, but let me sort of illustrate this. And I'm sorry, students, one more time. I have to ask you. Okay, so here's the PSA in abiraterone. Okay, so we have, here it comes at peak. You give abiraterone, it declines, and then it comes back up. Give it again. Now, if we're going to add docetaxel, cyclospecific chemotherapeutic drug, where would we add it in that cycle? So most people say here, right? Because you've got rising PSA, the tumor is growing, time to hit it hard. But I think the end time is here. Remembering that your goal here is not to kill as many cancer cells as possible. Your goal is to kill as many resistant cells as possible. And so the idea here is primary, secondary. If your primary therapy is abiraterone, then your secondary therapy should be used to kill the resistant cells to abiraterone. And where are those cells going to be proliferating? They're going to be proliferating, and those cells alone when abiraterone is present. And so what you'd want to do is give the dose of docetaxel here, and then when it's achieved this 50% decline, to get rid of both of those. Just don't treat it all. So you're not selecting for resistance to either one. Now the models suggest, in fact, that that strategy would triple the number of cycles that we would expect from this therapy. So again, just a way to sort of look at it. This has been sufficiently successful that we've now have a bunch of trials ongoing. They've actually been funded, and we're in the process of designing them. Each one requires enormous amounts of effort. So we're going to look at ovarian cancer, looking at combined cytotoxic drugs. We're going to look at targeted therapy with BRAF inhibitors. immunotherapy with HER2-positive breast cancer, first-line therapy in prostate cancer. So I'd like to propose that, well, cancer is complicated, and I think that often when you look at this vast literature on cancer, it seems incomprehensible almost. I would argue that we need to try to distill it to first principles. We need to distill it into sort of basic ideas, and I think that evolution can provide the fundamental principles, and that mathematics can allow those principles to be applied even to the complex dynamics of cancer biology and therapy. It adds nuance and I think context to the standard genetic models of cancer. And I think that it's likely that cancer therapy can benefit from the explicit inclusion of evolutionary dynamics to optimize currently available drugs. And with that, I'll thank the many people that have been involved and the funding that we received. So thank you. I'm just happy to answer questions. Yes.
Are we talking about the same thing from different languages, or are they really two different things?
It depends on what you mean by cancer stem cell, I think.
I don't personally believe it, but...
Okay. From an evolutionary point of view, the classic stem cell hypothesis of hierarchical distribution is sort of hard to imagine because it's very wasteful. You know, what you have is a lot of cells that end up not contributing to the long-term population. There's ways around that. So you could be constructing a niche, which then allows so it has to do with what's the unit of selection. Is it individual cell or is it this combination? My tendency is to think that model is not particularly good. However, the idea of stem-like phenotype and as part of the plasticity of cancer, I think would probably be an applicable language. And I think that we have to find sort of overlap. One of the things that we've done is taken the 4 molecular GBM subtypes, and we've looked at them we've characterized their adaptive strategies in this sort of way, so that we're trying to bridge those discussions. Yes. Right. Good.
So this is more of a philosophical question, but what is the definition of a species? So I presume in evolutionary biology, the definition of a species or a species in general is over time, both phenotype and genetic composition of that particular species and others. But in the way that so you propose at least 2 possible species within a tumor, and we arrived at a definition from evolutionary biology perspective, what constitutes a species and how many species can be defined in a tumor, and how it evolves.
Sure. So the question was, how do we define species? And it's a very good question, because in most organisms, it's whether they can breed and produce, live offspring, right? Because it's about sexual reproduction. Since cancers are asexually reproducing, that creates a significant problem in defining a species. So you're exactly right. The definition we use is that they breed true. By that I mean that the C2 and C3 cells, if you put them in a dish where they're not particularly being selected for anything, at the end of that, they still have the same properties in vivo as they had before. And so the fact that these 2 guys can maintain a different phenotype over when they're bred separately is what we've used for species. It may not be an ideal term. I mean, the evolutionary biologists use it, and it is a usable term in asexually reproducing species. But there's probably a better way to say this. But I think that it's important to think about, and maybe it's more important to think about niches. how many niches are there, and then maybe define populations by their particular niche adaptation. I think it's amazing that if you take a warbler species and you look at a pine tree, there are actually 4 warbler species in a single pine tree. That's how many. So it's assumed that there's these 4 niches, and so it's remarkable that even a single tree can have so many different niches. So you can imagine that a tumor does as well, but we don't think of it in those terms. So I think it's useful to at least try to define those. Yes.
So your mathematical model seems like reversal. Every time you can go back to the original state, right? So, for example, when you start treatment, and then your the cell, bad cell, good cell, always overgrow, right? That actually can your model be more reversible. So, but sometimes it's not. How do you treat like a reversible or evolutionary situation?
A reversible meaning that.
So your practice landscape from normal to cancer, sometimes after treatment, proper treatment, then you always get optimism that landscape come back to the normal landscape.
Oh, okay. Right? So what you're saying is the treatment could affect the landscape. It could, for example, change the blood vessels. Or the fibroblasts or the immune cells, so that the tumor itself becomes different over time. And I agree. I mean, I think those are the kinds of things that we'll have to think about. I think there's a couple of nuances to this, and one is, what is the evolutionary horizon of these cells? So if you have the sensitive cells in this region of the tumor and the resistant cells here, if they're not interacting with one another, the kind of dynamics that we've talked about won't be applicable. And we have to go to different models. So I wouldn't propose this as a sort of be-all and end-all models. It's really simply a first attempt and just kind of a toe in the water to see if we can at least not kill anybody with this. Because honestly, we had no idea when we started this. And in fact, our first 3 patients were, if we didn't get cycles in them, the protocol stopped. So there's a lot of worry about what was going to happen in this, and we didn't honestly know. So now at least have some level of confidence that we're not going to kill anybody. Yes.
This idea of hitting with the second line therapy while the first line is still responding before it starts taking off is interesting, and I think it might be sort of a hard sell for some people, but I can think of an example, I think, where it's been already in breast cancer for women with ER-positive, HER2-positive tumors, because they get chemo, they get anti-estrogen, they get Herceptin, and there's a set regimen to that. Like, you don't wait until the starts coming back up and it actually works pretty darn well. So,
Yeah.
would you see that as an example?
Yes, that does sound like that. We have to go through all the dynamics, but yes, it does sound like it was an inadvertent evolutionary informed model.
It's like you're hitting different populations also.
Yes. Yeah.
Go ahead. So I wonder what the role of lineage plasticity and the ability of a single cell to change lineage, what role that has, because in your definition of the species in the cancer cell is that it reads true, but in fact we know that cells can change their lineage without dividing, and so then when they divide, they're a different cell.
Yeah.
So how does that work in your...
Yeah, so we're just beginning to, so one of the, and I've talked with James quite a bit about this, is we're starting to look at more complex, more subtle variations over time. So what we've done here is the simplest fixed phenotypes that breed true. We're also going to be looking at phenotypic diversity, phenotypic plasticity, and how switching from phenotype to phenotype can occur, and how that can be exploited or how it can beat us. The fact that it changes does not necessarily make the cost different. So you may, in some ways, have the same dynamic. In other words, if you have a cell that doesn't express P-glycoprotein and then suddenly does, and expresses a lot, that cost is still there. So some of the dynamics are the same, but there is also a cost of transitioning, which is also interesting, and we've been thinking about that. But also the interactions of tumor cells and mesenchymal cells, and how that can provide an area of safety. And there's a lot of, this is really just sort of the beginnings of this, I think.
So I guess just to quickly follow up on that, is what role do you think that different molecular subtypes are going to contribute to that? So different molecular subtypes may make cells more plastic than others. And so what about incorporating that into the modeling?
Yes, it will have to be. So there's a couple of things. One is a generalist. There's costs and benefits to being a generalist. There's costs and benefits to being plastic, and the plasticity itself is probably a trait which can be selected or not selected. All of these things are part of the potential problems to be dealt with. The molecular properties, I think, are very interesting, and I just for a moment want to mention that how we sort of think about these things is that the molecular properties tell us the how, and they give us targets. They give us sort of quantitative metrics. So if we want to know how a cell has become resistant, we can look at the molecular properties of the cell. Whether that cell proliferates, or whether that population can then dominate the cell and progress, that's evolution. Those are Darwinian dynamics. So we tend to intellectually separate these two, but recognize that there's a lot of overlap, if that makes sense.
Thank you, Bob, one more time.